Hello, and how are you? Welcome to our session on cell organelles. We just began on the previous lessons with the cell membrane, and you've looked on the features of the cell membrane, the proteins and the lipids that are part of it, and we've seen the specialization that the cell membrane has, including the microvilli and the cell junctions. But in these lessons, we are going into details on the different organelles present in our cell and what are their different functions. If you're new here, my name is Winnie Barawa, and kindly remember to subscribe. If you're a returning subscriber, welcome home, have a seat, and be ready to jog in with me into the different organelles. So the cell organelles are quite many, and in our lessons, we're going to look at the 14 main cell organelles that are key and their functions. So we'll have all the way from the nucleus to the nucleolus, ribosomes, rough endoplasmic reticulum, vesicles, and we will finish up at the centrioles together with the smooth endoplasmic reticulums. Now, in the previous class when we started with the cell membrane, I explained how uniquely the cell is organized in such a way that it starts from the busiest region of the cell, going towards the least busy, but again, it goes towards areas that can transport the manufactured materials in our cell. So if you look at these cells, it's quite busy. But if you also feel confused, don't worry because we are going to discuss each and everything in here in its simplest way. So let's look at the central part, that is the purple part here. We are looking at the nucleus. And in the nucleus, it's covered by what we call the nuclear membrane or the nucleus envelope. And inside the nucleus, we have our nuclear materials, which is stored in form of chromatin. And at the central of it all, we have the nucleolus. Now, the nucleus, before we go into its functions, of course, we know it's for DNA purposes and facilitation of replication, protein synthesis, so many functions. Now, when we do these functions, we expect these materials, part of them to be transported outside. But you see, as you're coming outside here, you find the rough endoplasmic reticulum. There's a reason why it's organized like that, because we understand the rough endoplasmic reticulum manufactures proteins, packages them, and allows them to be transported to their required places. But now, if you have rough endoplasmic reticulum for protein synthesis, then it means you'll automatically need ribosomes because ribosomes actually have its own it has its more functions of also facilitating this protein synthesis. And so once the protein synthesis have been facilitated adequately enough, you can actually transport them to the Golgi apparatus. And these Golgi apparatus can pick them up. And what the Golgi apparatus do, they do packaging. They also check on the badly formulated proteins and destroy them. They also check on our lipids and all that. So the Golgi apparatus, as you can see, the green structure here, will package these materials or substances or the proteins or the lipids into vacuoles. And what the vacuoles will do, they will pinch off from our Golgi apparatus and head into the cell membranes for transportation in a process we call exocytosis. And this is how busy it looks. But you see, for the purpose of transporting materials, we need energy. And you know who you get here? The mitochondria. The mitochondria will be providing energy for the transportation of all these materials. And as you can see in the peripheral areas, we now have the lysosomes, whose main role is to destroy and produce lytic enzyme that can kill any microorganism. We got the centrioles here that will later help us with cell divisions. We have the smooth endoplasmic reticulum, which is closely together with the rough endoplasmic reticulum. This is also for the purpose of the lipids and so on. So let's keep in and now discuss this cell organelles one. Yeah, so we got it here again. We have our nuclear, so just by naming them, so we have our nucleus covered by the nuclear membrane. We have the nucleus itself containing all these materials, which are stored in form of the chromatin. And then we have the nucleolus, which is the stored nature of the DNA components, the nucleic components. And remember, the nucleus envelope has pores. Because it has pores, it means it can allow materials to cross in and out. Of course, it's also semi-permeable. It doesn't allow everything. It is selectively semi-permeable. and around it we have the rough endoplasmic reticulum, which has ribosomes on it, and then we have the Golgi apparatus, and the Golgi apparatus near to it, or rather Golgi apparatus produces for us what we call the vesicles or the vacuoles, and then we have peroxisomes, very important materials that help in destroying peroxides. Got microtubules that originate from the centrioles or the centrosomes. We have the mitochondria. Then we have the microfilaments, that is the tubules and the filaments. And then we have our microvilli on the cell surface for increasing surface area for absorptions and other reactions or functions. So we've got our lysosomes here. There's a cytosol or the cytoplasm that allows chemical reactions, and you have the smooth endoplasmic reticulum. Generally, these are the organelles that you're going to discuss on. So let's start with the first organelle, the nucleus. So we're told the nucleus contains the DNA materials or the chromosomes, which is stored in form of nucleolus. So the nucleus is enclosed by a porous double membrane known as the nuclear membrane or the nuclear envelope. And the main function of our nucleus is to control each and every function in the cell. So the main function of our nucleus is to control each and every function or everything in the cell. So just to describe it, this is literally the whole nucleus. The purple ball that you're seeing, whole nucleus, it has been cut cross-sectionally to expose the content in it. And so we have the nucleolus, which is the central part, and then we have the nuclear envelope that is covering it, and the nuclear envelope has the nuclear pore. These are chromatins, the threads of chromosome materials that have been stored that can later open up to allow DNA multiplication or DNA replications later on. Moving on, we have the nucleolus. So the structure of a nucleolus, it's a dense region inside the nucleus. It's a dense region inside the nucleus, and what it does is takes care of the RNAs and makes ribosomes. So it's the ribosome formation area, but it's also the RNA functioning area. So remember, we have DNA and then we have RNA. So the DNA and the RNA, these are the components that work closely to allow functions around replication, around protein synthesis. It generally controls the DNA functionalities. And for this purpose, the nucleolus takes control of that, but it's the central part of our nucleus. And the third organelle is the ribosomes. So the ribosome by structure, it's a tiny organelle that's made of proteins and RNAs. It's found on the rough endoplasmic reticulum, and what the ribosomes will do, they will function together with the rough endoplasmic reticulum in helping bring together amino acids to make proteins. So in short, the ribosomes, it's an area for protein synthesis. And when you come to discuss protein synthesis, you will see how the ribosomes come together for this. In here, there are also ribosomal RNAs that work together to facilitate all this protein synthesis. So yes, there are structures found on the rough endoplasmic reticulum, and they work together with the RNAs to help us create proteins or manufacture proteins. The fourth is the rough endoplasmic reticulum. And the rough endoplasmic reticulum, this is a tiny folded membranes that are connected together. They have ribosomes, and the main function is to transport materials. It allows a pathway for transportation of materials. So the ribosomes are these plates here, the tiny folded membranes that form together plates. And these plates are like channels of transportation. So they allow materials to be transported here, primarily proteins. So just remember, this is primarily proteins. And as because it's a highway, as materials are transported across here, it also allows it to be packaged. So proteins are the main key area, and in this place, we allow more protein synthesis. They are also able to make their own more endoplasmic reticulums that can be used for further protein synthesis. And just to remind you, they have ribosomes. And these are the small tiny dots that you're seeing here at the ribosomes. Fifth is the vesicles. So the vesicles, in short, we can call them the transporters or the transporters of the cell. So the vesicles can be of different nature, but the main thing is these are membrane-bound containers that allows us to carry substances throughout the cell. They allow transportation of materials across the cell. If you need to take in manufactured proteins from one point to another, you can use the vesicle for that purpose. If you want to take in lipids from one area to another, the vesicles do that work. So what the vesicles do is mostly they pinch off from Golgi apparatus, or they tend to originate from the rough endoplasmic reticulum and get to be transported to the different parts. But the main thing is they allow packaging and transportation of these materials. So like here you have the Golgi apparatus packaging these components, and then they normally pinch off as the vesicle. So you can have vesicle that are of transport vesicle like here. So what the transport vesicle are, they take proteins to the Golgi apparatus. So here we can say they originate from the rough endoplasmic reticulum, which has synthesized proteins and packaged them in the vesicles, and they will direct them to the Golgi apparatus. So once they arrive on the Golgi apparatus, the transport vesicle will attach to our Golgi apparatus and release the materials into the Golgi apparatus. And so when the Golgi apparatus are done cleaning and modifying either the lipids of proteins from the ER, they will now package them again into new vesicles, which at this point we are going to call them the secretory vesicles. So they will transport our materials to the cell membrane, attach, and release them to the external environment for their use. And as you can see, they will package different things. You cannot mix two components at the same time. So it means they have to be specialized. So if you're transporting proteins, then you'll need to use separate vesicles on their own. If you're transporting lipids, you have to use separate vesicle on their own. And you're able to take all these to the Golgi apparatus for cleansing or modification and removing any worn out or improperly prepared materials, and then you can release them. Note again, there are other vesicles coming in. These are the incoming. So here they can also receive components from outside and bringing in into the cell where they are needed. So yeah, vesicles are the busiest transporters for our cell because they allow materials in and out. Sixth apparatus, Golgi apparatus. The Golgi apparatus, it's a structure of closely layered stacks of membrane that are enclosed, the disc or the enclosed looking like discs. And the main function of our Golgi apparatus is to do checking, modification, and packaging of proteins. So with the Golgi apparatus, like the way it says, it's a stack of layers. And because it's a stack of layers, it creates membranes or it creates discs. And because it creates a disc-like appearance, it comes in with face. So the Golgi apparatus has a cis face and then it has a trans face. So just to bring you in so that you can see. So it has a trans face and it has a cis face. So with these names, the cis face means incoming or the entry point. The trans face is for the name transport or exit point. So remember the Golgi apparatus is for checking and modification of our lipids and proteins. So what happens is these proteins and lipids are coming from the manufactured points. And these manufactured points are the rough endoplasmic reticulum and the smooth endoplasmic reticulums. So when the materials come on the cis phase, they will be coming in using the transport vesicles or the incoming transport vesicles. So these vesicles will come, attach on our cis phase of the Golgi apparatus and empty the content there. Once they empty the content, our Golgi apparatus using this cysternize in the lumens here, they will cleanse, they will destroy any unneeded contents, they will remove all the bad parts. They will remove all the bad things that are in the proteins and lipids. And then once it's done, they will repackage our materials again into new secretory vesicles. And then they will pinch off, but they will not pinch on the cis phase. They will pinch off from the trans phase. So on the trans phase or the transportation phase, they will release the new vesicle or secretory vesicle, and therefore they will transport these materials to the other parts of the cells. But now to just focus on the Golgi apparatus, this is how it looks. Has a cis phase, has a trans phase, layers itself to give in stacks or to create the discs, and this is what gives the appearance of a Golgi apparatus. So moving on, here is our Golgi apparatus again. It's among the busy organelles also. So the process of how molecules behave, or what I was describing prior, is this. The molecules come in using vesicles, and they fuse with the Golgi apparatus on the cis phase. And then the molecules will be modified by the Golgi apparatus. And once the modification has happened, all their needed areas have been removed, the molecules have been modified successfully, then they will pinch off into separate vesicles, what you are calling the transportation vesicles. And then these vesicles will leave the Golgi apparatus, and the vesicles will head all the way to the cell membrane. They will attach there and secrete the content. Okay, and so here is the whole process that is happening. So the cis phase will receive vesicles that are originating from the rough endoplasmic reticulum, and they will come on the cis phase. They will attach here, empty the content into the Golgi apparatus cis area. The materials will get to be modified or cleansed. All the bad materials will be cleaned inside the cystinize. I hope you're seeing this, the cystinize. They will clean all these materials within these channels here, making sure everything is cleansed. And once that work has successfully been done, we will form new vesicles that will pinch off from the trans phase. The trans phase is, like we're saying, is the transport side of the Golgi apparatus, or the shipping side of the Golgi apparatus. So these new vesicles, we will be calling them the secretory vesicles, or again, you can still call them the transport vesicle. They will pinch off, and now they will start the journey of transporting the materials to the needed parts of the cell. Could be towards the cell membrane or the plasma membrane, where they will attach or fuse, and then they will open up and release the materials to the outside of the cell, okay, in a process we call exocytosis. All right, and because they can perform exocytosis, so our materials will now, so this is our Golgi apparatus, just explaining it in more details. So we have the rough endoplasmic reticulum releasing vesicles, and this vesicle from the endoplasmic reticulum will come to the cis phase of the Golgi apparatus, attach, release the materials, be cleansed, and then they pinch off again here. Or you can have new pinching all the way from the trans phases, and they can be transported to other parts of the cells or outside the cell as attaching at the plasma membrane through exocytosis. Very busy organelle. So all these are more explanations, and this is how the linking happens. So you have the rough endoplasmic reticulum here. The rough endoplasmic reticulum also has cysteinized. It has manufactured proteins. They are pinching off, and they are trying to look for our Golgi apparatus. So they'll come, attach on the Golgi apparatus, or rather the vacuoles from the rough endoplasmic reticulum will pinch off, attach on the Golgi apparatus. The materials get to be modified or cleansed, and then they pinch off as the vacuoles or the vesicles. And so they will move out, attach on the cell membranes, and allow materials to be exhausted or to be removed to the outside of the cell. And this secretion model, we call it exocytosis. So these are all vacuoles coming all the way from the Golgi apparatus. And they can even participate in phagocytosis if they realize they are further harmful or toxic materials, the lysosomes can also attach to it, release lytic enzymes, and destroy the needed toxins. So thank you so much. And like I said, if this is your area of interest, remember to give us a subscribe. Just click on the bell-like area to get notified also when we post our next class. So thank you for staying with me to the end. See you in our next lessons. Bye bye.